Hi everybody, welcome to another Chats with Chaudhry. It's slightly different because we're here at the Antibody Therapeutics and Manufacturing Summit, and we're at the Bruker Daltonics stand, and we're actually going to get a poster presentation on, and wait for it, this is quite interesting, what if all 6 infinity stones made an SPR instrument, the Pro Series instruments. And actually, we're not talking about the Marvel Infinity Stones, but Sven Malik and Cyril Brunner are going to tell us more. So, gentlemen, how are you?
Good.
Fine, thank you.
Excellent. So, Sven, if you'd like to talk us through the poster, that'd be brilliant.
Absolutely. So let me share my screen, and then we just, so you should see that now, right?
Yep.
So actually, this is the poster. And our plan was just to give you an idea of that. So as you mentioned already, the title. So what if all 6 affinity stones made an SPR instrument, the Pro Series instruments? So in April 2020, we launched the SPR 24 Pro, and a little later the 32 Pro, which is our latest SPR instruments at the Bruker. At Bruker Daltonics SPR, and we thought, which are the key parameters for creating a powerful and then a really good SPR instrument? And these are the 6 infinity stones that you can read here, which is sensitivity, flexibility, empowerment, throughput, reliability, and efficiency. And the SPR 24 Pro and also the 32 Pro consist of 8 channels, which you can see here. And this is an example here for the 24 Pro, and within each channel you have 3 sensor spots that can be individually addressed. And we laid out here a workflow for the therapeutic screening, which will Cyril go over a little bit later. But yeah, the system really includes a lot of flexibility, sensitivity, and finally also throughput. So this is what I would like to mention here. You either can use the system as a single needle system, or if you go to the high-end throughput, you can connect it to an external robot and also integrate it into an fully automated environment via its API, so its application programming interface, which is really nice. So yeah, that's actually the outline of the poster, and that's the idea behind it.
Fantastic. Yeah. So, Cyril, can you tell us a bit more about the applications and the workflow?
Yeah, so the Pro Series comes with a whole broad bandwidth of features that can be used for applications. On the one hand, I've heard already from Sven, it has this 8 needle design, so it has a high throughput, allowing you to screen and measure as many samples as possible in a fast way. On the other hand, with the 3 or 4 measurement spots, depending on which system you have, you can go with up to 31 antibodies immobilized on your sensor chip and measure it in one shot against one antigen. Then we have features included which allow us to do an epitope binning mapping experiment. You can easily analyze the interaction of, for instance, an antibody with FcRn, so with a neonatal receptor at different pH, all in one shot. And also, if you have those typically difficult, slowly dissociating antibodies from your surface, go just to 37 degrees, our machine can handle that easily. I have here an example of such an application, of such a typical workflow that you would go through in biotherapeutics. So you have your cells, your hybridoma cells expressing your antibodies. You first do, as shown here in picture 2A, concentration analysis. You determine what is the concentration of your antibody in those samples. It's a qualitative statement, but on the other hand, also a quantitative statement of the expression of your antibodies. Then you go over to the screening. We did here a screening, as I described before, where you have a capture surface on your sensor chip. You capture 31 antibodies, or in that case, we took 24 antibodies. You capture them on the sensor surface, flow over your antigen, strip them off, both of them again, code new, and so on and so on. And in that way, we screened a library of 96 antibodies. we got 21 hits, meaning 21 antibodies which did show a response. And out of those 21 hits, we further characterized based upon concentration, sort of a mini profile in terms of kinetics, because that is the big advantage of SPR in comparison to methods like ELISA, where you only get binding or no binding to some extent, but you don't get any information on the kinetics, which is equally important because you want to know not only how good they bound, but also how long they actually stick to the target. With that, we got then the 14 leads, which we further characterized here in figure 2C in terms of their kinetics, and got some really good binders. Eventually, one thing that you always want to look at is the better understanding of the binding. And epitope bidding mapping is one of those tasks that you have to do, where you want to figure out whether your antibodies are all binding to the same epitope of your antigen, or whether they are binding to different ones. Also an important aspect if you want to go around certain patterns where you know, okay, this antibody is binding an antigen at that position, let's make an antibody which is binding at a separate position. To do that, we actually need a system which is highly sensitive, which you see in figure 4. Our system is able to easily go also in the range of 0.5 to 1 RUs, which is necessary if you actually want to resolve interactions that are super strong. So highly affine interactions, nanomolar, picomolar range, where you are afraid of mass transfer limitation, then you need that type of sensitivity. Also, you need a highly reliable system. That is on figure 3. Our system comes with a novel microfluidic that is, on the one hand, valveless, but also much more robust. It allows you to not only measure basically all types of sample, going from purified samples all the way to crude samples. It also allows you to address each and every spot individually, and the system is also, with the sensitivity combined with the sensitivity, easily capable of going for also those very difficult binders, those antibodies, picomolar affinity antibodies, where you need low immobilization levels, where you need a good sensitivity, you need high flow rates. We can easily go up to 100 microliters per minute, and you need also sometimes temperature, because you just don't get rid of them over a long run. So that's what we have it as well.
Brilliant. So in conclusion?
In conclusion, our Pro Series is a combination of state-of-the-art in sensitivity, flexibility, especially throughput. It's the highest throughput on the whole market, and a rich arsenal of features, which give you as a user the possibility to combine as many things that you need in one single machine, going from assay development all the way to screening, and then eventually determination of what you want to know. Yes, and with that, we also would like to acknowledge In Vivo Biotech, who actually gave us the samples for this nice study that we have here as an application case. And well, thank you.
Brilliant.
Yeah.
Thank you very much.
Thank you,
Stan and Cyril, for going through that with me. Very interesting. So I hope you found that useful. If you'd like to know more from the team at Bruker Daltonics SPR, make sure you go to their booth. They've got lots of other information in there as well, and have a talk to the team there. So as always, gentlemen, thank you very much for taking the time out to go through the poster presentation.
you're welcome.
And Sven, I have to compliment you on that lovely cactus behind you as well. It's,
Yeah.
yeah. Or both of them, actually. I didn't see the
Yeah,
one on the
yeah, the white one is difficult to see. Yeah.
So, Cyril, lovely to meet you. So, lovely to meet you both. Thank you for doing the poster presentation. As I say, please check out the Bruker Daltonic SPR booth, find out more, and have a talk to the team. They'll be there the whole week. So thank you very much, gentlemen. Until next time, as always, stay well and stay safe. Bye-bye.
Bye.
Bye. Thank you.